The overall goal of the following experiment is to fabricate biodegradable fibrous meshes and nano or microparticle coatings from blends of biomedical polymers and to tune the wettability of these materials by varying the copolymer blend. This is achieved by first synthesizing functionalizable copolymers in a ring-opening polymerization and subsequently grafting on hydrocarbon C18 chains. As a second step, the copolymers are dissolved along with polycaprolactone or polylactide coglycolide in an organic solvent and subsequently electrospun or electrosprayed, which results in three-dimensional microfiber meshes or nanomicroparticle coatings, respectively. Contact angle goniometry studies are then performed on the meshes and coatings in order to understand how fiber diameter, copolymer composition, and blend composition affect surface wettability and lead to superhydrophobic biomaterials. The results show how hydrophobicity is enhanced by increasing the concentration of copolymer in the electrospun blend, as determined by advancing and receding water contact angle studies.
The implications of this technique extend towards situations such as chemotherapy because these materials can serve as local drug delivery depots for sustained release of entrapped agents, with the relative drug release being controllable by polymer composition and surface roughness.
To begin, synthesize the monomer required for the experiment by following the instructions in the accompanying text protocol. Then, preheat a silicone oil bath to 140 degrees Celsius. Measure out 2 portions, 2.1 grams each, of 5-benzyloxy 1,3-dioxan-2-one and add it to separate, dry 100 ml round-bottom flasks. Next, measure out 5.7 grams of DL-lactide.